Evolution results from changes in DNA. These changes are caused by random mutations, changes in the DNA sequence. Mutations are often detrimental, but some mutations are neutral. Neutral mutations neither benefit nor decrease an organism's ability to survive and reproduce. Neutral mutations are evolutionary changes and occur in a clock-like fashion. They act as a sort of molecular clock. Molecular clock is a technique used in evolutionary biology that estimates the age divergence of organisms from a common ancestor by looking at differences in their DNA. Scientists measure the differences between the DNA in certain genes. Often, mitochondrial DNA of organisms is used because its rate of mutation is well established. They use this molecular clock to determine when organisms diverged from a common ancestor. In this example, two different species had a common ancestor sometime in the past. To determine when they diverged from a common ancestor, scientists use a molecular clock based on a gene that they know changes by 2 bases every 25 million years. They observe that the species differ in the DNA of this molecular clock gene by 4 bases. If the species differ by 4 bases, they must have diverged from a common ancestor around 50 million years ago. It's difficult to calibrate the molecular clock against actual time because the rate of mutation depends on factors such as generation time and population size. The calibration leads to some uncertainty in dates of divergence. However, using multiple genes along with fossil data can help narrow down the dates. Scientists have identified genes that have more constant rates of change than others. These make the best molecular clocks. Biologists have used the molecular clock technique to determine the date of the divergence of humans and chimpanzees. The data show that the last common ancestor of chimps and humans lived around 5 to 7 million years ago. This ancestor evolved into various hominid species, one of which is an ancestor of modern man.